Hey, Mr. P here. In this video, we're going to talk about how peas are crossed, how Mendel used peas, why Mendel used pea plants, and how he crossed and ran his experiments in order to study single gene traits. We call single gene traits monogenic traits because they are traits that are controlled by a single gene. Obviously, a single gene trait is going to be determined by the presence of a dominant or recessive allele. We know at this point in our genetic kind of education or our genetics understanding that a gene is going to be a discrete unit that is inherited from parents to offspring. Those genes are going to code for traits, and the gene is going to be a combination of two distinct, possibly different alleles. You get one allele from mom, one allele from dad. So this is Crossing Pea Plants, Mendel's study of single gene traits. Let's go. The first thing that I like to talk about is why peas, okay? But in order to understand why peas, I want to start with a quote from Gregor Mendel himself. And he said, "The value and validity of any experiment are determined by the suitability of the means as well as by the way they are applied. In the present case, as well, it cannot be unimportant which plant species were chosen for the experiments and how these were carried out. Selection of the plant group for experiments of this kind must be made with the greatest possible care if one does not want to jeopardize all possibility of success from the very outset." This was a specific quote that he put in his background kind of introduction for his entirety of his experimental work. And he knew that the success at the end, or the possibility of success at the end, was vital to the correct identification of a legit plant species that would give him, in his mind, ultimately the success that he was looking for. And he was looking for how are traits inherited, and were there ways in order to predict the inheritance of traits, and what are the ratios, or what is the probability of certain traits being inherited from parents to offspring. And so he chose pea plants, and it wasn't just a random selection. He chose pea plants on purpose because he knew that the proper plant species had to be chosen in order to give him the greatest possible success. And he did find much success, and much of his success was because of the fact that he chose pea plants from the onset. So, why peas? Peas had many of the properties that Mendel was looking for in an experimental organism. And so what are the properties that he was looking for in an experimental organism? There are 30 varieties plus, but 30 different varieties with different traits that were available from seed dealers at the time. And not only were there a variety of seed varieties, the seed varieties exhibited a great diversity of very easily identifiable traits. When you looked at peas, there were peas that differed in their seed shape. Okay, we call them round and wrinkled. It is very easy to see when you're looking at peas that were produced whether or not they exhibit the round trait or the wrinkled trait. Seed color, yellow or green. Flower color, purple or white. Pod shape, inflated or constricted. Pod color, yellow or green. Again, all of these, very easy to identify. Flower position, they're either axial or they're terminal. Axial means they're halfway down the plant. Terminal means they flower on the very tips. Stem height, you have tall plants and you have dwarf plants. Again, very easy to tell whether or not this particular pea plant is exhibiting the tall trait or the dwarf trait. Now, all of these are really, really easily recognizable traits. And so he then needed to look at, well, this organism checks off the box for being or possessing a variety of traits that are easily identifiable. But now, how viable is the crossing mechanisms, or the artificially fertilized, or the artificial fertilization protocols? And so the plants that he looked at, all of these plants that exhibited these traits, can be self-fertilized, meaning they can be fertilized using normal plant reproductive processes, or just as easily artificially fertilized by hand. He could control the crossing by taking pollen from one plant that exhibited one trait and putting the pollen on the female parts of the other plant that exhibited different traits. And so he could very easily control the crossings, which is important in genetic testing. Okay, and when he looked at the crossing of these traits, all of the offspring are fully fertile, which is an important trait too, because a really important property, because if you make offspring that are infertile, it does you no good from a heredity or a genetic standpoint, because you can't take that plant to further generations. Peas have a relatively short life cycle, like we talked about in the last video. They can be grown outside, they can be grown in a greenhouse, they don't take up much room, they don't move, they have very easy housing requirements, okay? They just need water and sunlight and ample space, but the space that they require doesn't take up much space. And so when he kind of looked at all of these different properties, he determined that peas were the perfect organism for him. So for his experiments, Mendel chose 7 different varieties of pea plants, and each had a distinguishing characteristic called a trait. Seed shape, trait 1, okay, or variety 1. Seed color, number 2. Flower color, 3. Pod shape, 4. Pod color, 5. Flower position, 6. Stem height, 7. For all of his experiments, he used these 7 different varieties of pea plants, and all of them had a different characteristic called traits. And so this was kind of the foundation, and those that we just talked about are the reasons for why he chose these plants. Each trait was represented by 2 variations. You can see that there is basically a dominant variation in this particular column, and you have a recessive variation, which can be seen in these columns. Okay, so for every variation, like seed shape, there was a dominant trait and a recessive variation. Okay, so crossing pea plants, looking at how he utilized single gene traits in order to set up his crosses. The way he set up his crosses was he always looked at a trait independent of all the other traits. So for this particular topic or this particular slide, we're using seed shape as the variation that we're looking at. In his P generation, which stands for parental, he always crossed a dominant and a recessive form of that particular trait. Now, if he was looking at flower height, obviously he would use a dominant, the tall, and the recessive, the dwarf. Okay, but like I said, for this slide, we're just focusing on seed shape. But again, seed shape is not the only variation or the only variety he was studying. In making the cross, flowers from one variety were fertilized using pollen from the other variety. So let's just assume that this is the pollen. Okay, he took the pollen from a dominant round seed plant or round pea plant, and he crossed it with the female ovule from a recessive plant or a plant that exhibits the constricted or the wrinkled pea shape. Okay, took pollen from this plant, gave it to this plant, and so he crossed this plant with this plant. When he did that, the peas that formed in the F1, which stands for first filial, okay, F1 stands for first filial, it's the first offspring generation, were all smooth. So they all looked like the original father. Were they exactly like the original father? Well, he needed to find out. So the way he did that was he then self-crossed the F1 plants with the F1 plants. And so he self-crossed, meaning this is what that arrow is for. Okay, he crossed an F1 with an F1. So he took an F1 male, crossed it with an F1 female, and he found that out of 7,324 peas that were produced as F2 offspring, which by the way stands for second filial, just like F1 was first filial, F2 is second filial, he got roughly a 3-to-1 offspring ratio in the F2 because the results showed that out of 7,300 pea plants that were produced, roughly 5,400 resulted in the smooth phenotype, and 1850 resulted in the wrinkled phenotype. So when you take the ratio of those two, it gives you roughly 3 to 1. Okay? Why is that 3 to 1 important? We'll talk about it in a minute. So, in all cases, in all 7 varieties that he studied, and 7 varieties of peas that he looked at, he drew some conclusions from his first series of crosses. Only one of the parental traits were present in the F1 always, so it didn't matter if you crossed seed shape or seed color. All of the F1s, the first filial offspring, looked like the original parent that exhibited the dominant trait for that particular variety. The trait that wasn't present in the F1 reappeared in the F2, but only in roughly 25% of the F2 plants, meaning it never became the majority. It always kind of was left as the minority, right? So that, again, plays into that whole 3 to 1 ratio. 3 to 1, meaning 75% shows the dominant phenotype and 25% shows the recessive phenotype. And in all cases, or in all crosses, it didn't matter which trait was present in the plant that contributed the pollen, the results were always the same. And so what I mean by that is it didn't matter if the pollen came from the male, or the pollen did come from the male, but it didn't matter if the pollen came from the dominant or if the pollen came from the recessive. It didn't matter if the egg came from the dominant. It didn't matter if the egg came from the recessive, okay? It did not matter which gender, male or female, contributed the dominant trait. The dominant trait, regardless of gender and regardless of parent, always shown up or was expressed more often than the recessive. Okay? So, all 7 traits were the same as those seen with smooth and wrinkled peas. He did all of the same type of experimentation I just talked about for this particular variety, and for all of the varieties that he tested. All of them came out to be roughly 75% dominant, 25% recessive, or that 3 to 1 ratio, which is now known as a Mendelian ratio. The results of these experiments were Mendel's first discoveries. Obviously, like I said, now it is a Mendelian ratio, 3 to 1, for monohybrid crosses. Monohybrid meaning a cross between one or a single gene trait. He concluded that traits were not blended. As they passed from parent to offspring, they remained unchanged, even though they might not be expressed in a specific generation. Again, when he took from parent to F1 to F2, they were not expressed in a specific generation. In the F1, right, all of these recessive traits are always masked, right? They're always not expressed in the F1, but they always reappear in the F2, because in the F2 we have a 25% chance of our little or recessive alleles assorting out and being inherited together, which gives rise to the recessive trait. Okay? Instead, he concluded that traits were inherited as if they were separate units, separate genes that did not blend together. In all of his experiments, it did not matter whether they were male or female plants. In the P1, the results in the F2 generation were the same. Again, like I said before, it didn't matter if the dad, it didn't matter if the plant, the offspring plant inherited the dominant from the mom or dad. It didn't matter if the offspring plant inherited the recessive allele from the mom or dad. All of the results are the same. Okay? He concluded that each parent made an equal contribution, equal being the important part, to the genetic makeup of the offspring. Okay? Parents, male and females, moms and dads, give an equal inheritance to each of their offspring. Based on the results of his crosses with each of the 7 characteristics, Mendel came to several conclusions. 1, genes determine traits and can be present but not expressed. Again, some genes are masked, right? Obviously, these recessive traits are masked in the F1. Dominant traits are phenotypically expressed in the F1. These are traits that are controlled by dominant alleles, or big uppercase letters. Recessive traits are present but not expressed in the F1, however reappear in the F2 when the individual is homozygous recessive. Again, little letters are going to be expressed as the recessive trait when they are expressed together. Dominant traits, that is the uppercase letter. Because both smooth and wrinkled peas are present in the F2, Mendel reasoned that the F1 plants must carry genes for smooth and wrinkled, and so they do. Okay, and so what this means is the dominant plant in the P generation must have been big A, the recessive plant must have been little a, the little letters give rise to the recessive trait. Okay, in this case, it would be the wrinkled P. The dominant letters give rise to the dominant phenotype, which would be the round P. When these individuals give their offspring, the F1, their genes, the offspring will always be heterozygous, meaning they get the big A from their dad, let's say, and the little a from their mom. And so they are all heterozygous, meaning they all possess this big A allele, which means all of them, 100%, are going to be the dominant phenotype because they all express this big A as the dominant phenotype. And then when these are self-crossed, now all of a sudden they give rise to 3/4 of their offspring in the F2 being big A something, and by big A something I mean some of them end up being big, but most of them end up being heterozygous just like their parents. But 25% of the offspring end up getting the little a, which means this recessive phenotype, in this case wrinkled, shows back up just like it did with the original P1 mom in this case. Okay, again 75% because you have ultimately more options that give rise to this dominant phenotype, 25% because there is only one genotypic option that gives rise to this recessive phenotype. Okay, the simplest explanation is that each F1 plant must carry 2 genes for seed shape, one for smooth, the other for wrinkled. Okay. Again, F1 plants, they carry the dominant for smooth. They also carry the recessive for wrinkled. And then depending on how those alleles assort gives rise to the different F2 genotypes, which ultimately gives rise to the different F2 phenotypes. Okay, but if a parent plant has 2 copies of the gene for seed shape, why don't the offspring have 4, right? It's basically saying if this original parent has 2 copies and this original parent has 2 copies, why doesn't this individual have 4 copies? Why doesn't this individual get both big A's from this parent and both little a's from this parent? Okay, we know from our understanding of meiosis that in order to produce gametes, we actually have to cut the number of chromosomes in half, meaning we're going to split these to produce the gametes. And so the gametes are haploid, meaning they're only going to possess 1 copy of our chromosomes. He didn't know about that at the time, so this was kind of a big question mark that he then had to further study in order to kind of draw conclusions about how traits are inherited. And another question is, why doesn't the gene number double in each succeeding generation? Meaning, if this gene or this offspring or this individual has 2 genes, and this one would get 4, right, like he thought with his original question, then why doesn't this one have 8, right? Because if you're crossing 2 individuals each with 4 chromosomes or 4 copies of chromosomes, then these should have 8, and then the next generation, the F3, should have 16, and so on. Okay, so 2 big questions that he then had to find the answers for. Obviously, one of them, why don't the gene numbers copy from parent to offspring, and why don't the gene numbers double from parents to offspring? Okay. Although the P1 and F1 plants were identical in appearance, Mendel concluded that they were genetically different. Again, we've kind of gone through that, right? These are, we know now, to be different genotypically. He didn't know they were genotypically different just by looking at them, because the appearance gives you the appearance that they're genetically identical because they look genetically identical. And so that gives rise to some other kind of understanding, some other vocab terms for this particular unit. Phenotype, what an organism looks like. That's the physical expression. This P1 organism looks just like this organism, and those organisms look just like this one. They all have the same phenotype. Do they have the same genotype? No. The genetic makeup of an organism, the actual letters, okay? Phenotype is the physical expression. It's the looks. Genotype is the genetic makeup, so the actual letters, okay? Big A and big A little a are genotypically different, but these genotypes give rise to the same phenotype. In this case, they would both be round. More terms would be alleles. These are the variations of a specific gene. Okay, the alleles are the different forms of a gene. If this gene is P shape, we have two alleles. We have a dominant allele and we have a recessive allele. Okay, the alleles, based on how they assort and how they come together, gives rise to the genotype, which ultimately gives rise to the phenotype. And homozygous is an organism that carries identical alleles of a gene. So homozygous dominant would be big A, homozygous recessive would be little a, and then heterozygous is when an organism carries non-identical alleles of a gene, which would be big A, little a. Okay, now I'm using A's for all of this. Don't think that genetic crosses always use A's. I like A's because it's very easy to tell the difference between a dominant A and a recessive A, but both of them do play a role, right? Big letters are dominant, little letters are recessive. Bring your questions to class. See ya.